When we discussed the modes of action, last lecture, I brought this up in a roundabout way and tried to discuss it. When we're talking modes of action for these chemicals, these compounds, what they refer to as the antimicrobial agents, we want to know exactly the target and exactly what chemical reactions occurring on or to the target. So we find the cellular targets for microbes. We want to try for most often to go for things that, we don't have. Great one we're going to find for antibacterials, the antibiotics, is number one here in this list, cell wall. No place in your body do you have a cell wall, but most microbes do. Now, it may be thick, like gram-positive cells, or it may be thin, like gram-negative, but you interfere with it. You interfere with the ability of that bacterial cell to maintain its shape, to maintain its integrity. Another one is to go for the cytoplasmic membrane. You interfere with the membrane, interfere with membrane potentials, interfere with the ability to transport things across the membrane. Better yet, you interfere with integrity of the membrane. Remember that the whole point of a cytoplasmic membrane is to be a boundary. Inside, outside, outside, inside. Keep things in, keep things out. You interfere with the integrity, stuff leaks in, stuff leaks out. Or you go for the DNA or the RNA. You interfere with the processes of gene expression. You interfere with the process of DNA repair. You interfere, a great one when it comes to viruses. Remember, there are RNA viruses. You interfere with the DNA-dependent RNA polymerase. DNA-dependent RNA polymerase. Nowhere in your body, RNA-dependent DNA polymerase. That's better. Nowhere in your body do you have reverse transcriptase. Perfect target. Then you also go and you interfere with the proteins, interfere with enzymes. Maybe there's a enzyme pathway you can interfere with that will, mess up, slow down microbial metabolism. Folic acid synthesis is great for this. Sulfa drugs, one of the original antibiotics, antibacterials that were developed. Sulfa drugs interfere with folic acid synthesis. We humans don't have folic acid synthesis. Folic acid is something that we have to take up in our diet. A lot of microbes synthesize it themselves. Perfect target. So when you go and you look at this, table 9.3 here, you see that there are going to be different agents that can be used, and the effects these agents are going to have will be different. You look at cytoplasmic membrane, great agent for against this. Detergents, the stuff in our soaps. Turns out that the surfactant molecules that are in detergents that help, literally make the bubbles. What they do is they insert into the membranes. Turns out they are, amphiphilic. They are both hydrophobic and hydrophilic, quite similar to a phospholipid. So as you see here, they insert into the membrane. They insert at high enough levels, high enough concentrations into the membranes, they interfere with integrity. Stuff diffuses in, stuff diffuses out. The cell cannot maintain a metabolism when all of its internal stuff are leaking out. A lot of times students ask me, well, why doesn't this damage us when you're washing your hands? Well, technically it does, but the outer layers of the epidermis are dead skin cells anyways. They're full of keratin. There's no metabolism going on there anyways. So you damage those dead cells and you're sloughing them off anyways within a few minutes or as you're washing hands, so it doesn't matter. Better to lose a few thousand skin cells and the millions and millions of bacteria that are on your hands than risk having an infection.